Welcome everyone. I would like to present a brief overview of the findings from this project based on the abstract for the Dissolution research presentation entitled Microencapsulation Study of PLGA-Based Leuprolide Acetate for Sustained Release Drug Delivery and In Vitro Characterization. As an introduction, this research is motivated by the relatively high prevalence of endometriosis among Asian women, which is significant considering that Asia accounts for about 60% of the global population. The Asian Society of Endometriosis and Adenomyosis reports a prevalence of approximately 10% among Asian women of reproductive age. One therapeutic approach involves the use of leuprolide acetate as the active pharmaceutical ingredient. However, leuprolide acetate has a short half-life of about 3 hours. To ensure consistent drug release with extended dosing intervals, a sustained-release drug delivery system with a therapeutic duration of several weeks or months is required, such as microspheres. Microspheres release active agents through several mechanisms, including diffusion through the pore network, diffusion across the microsphere coating, and hydrolysis and erosion of the polymer. Drug release profiles from microspheres can generally be classified into three types, with the type 1 is initial burst release at low levels, followed by a lag phase and then sustained release. Type 2 is initial burst release at low levels, followed directly by sustained release without a lag phase. And then type 3 is initial burst release at high levels, followed by sustained release. These release profiles are influenced by several factors, including particle size, porosity, and in vitro testing conditions, which became the optimization parameters in this study. The objectives of this research are to develop PLGA-based microspheres for sustained release of leuprolide acetate using the double emulsion solvent evaporation method, to characterize the microsphere's morphology, thermal profile, and in vitro dissolution behavior, and to compare the formulation's release performance and stability with a reference product under accelerated conditions. This illustrates the microsphere synthesis method. In this study, microsphere were synthesized using the double emulsion solvent evaporation technique as leuprolide acetate is a hydrophilic substance. The primary aqueous phase consisted of leuprolide acetate and gelatin. The organic phase consisted of PLGA and dichloromethane, and the secondary aqueous phase was a PVA solution. After emulsification, microspheres were collected, filtered, and lyophilized by freeze drying. The final microspheres were characterized using Fourier transform infrared spectroscopy, thermal gravimetric analysis, differential scanning calorimetry, particle size analyzer, and scanning electron microscope. In vitro dissolution testing in this research was performed under two different conditions. The first one is using an orbital shaker with 10 milliliters of medium at 175 rpm, and the second condition is using a matrix orbital with 1 milliliter of medium at 300 rpm. The results of particle size evaluation of blank microspheres with varying PLGA concentrations and emulsification speeds showed that particle size distribution increased with higher PLGA concentrations under the same processing conditions, particularly at D50 and D90. This is due to increased viscosity of the water in oil emulsion at higher PLGA concentrations. Compared to the reference product, blank microspheres exhibited smaller particle sizes. In contrast, increasing emulsification speed had little effect on particle size distribution at constant PLGA concentration. Under the same equipment and process conditions, we know that particle size was more strongly influenced by PLGA concentration in dichloromethane. Morphological evaluation of blank microspheres showed that at the same emulsification speed, porosity was first observed at a PLGA concentration of 500 mg per milliliter. At the same PLGA concentration, porosity began to form at emulsification speeds of 15,000 and 20,000 rpm respectively. These are the differential scanning calorimetry that showed similar onset and onset values for all formulations and pure PLGA. The glass transition temperature of blank microspheres remain unchanged compared to pure PLGA, indicating that there is no change in the thermal characteristics of PLGA after microsphere formation. These are the thermal gravimetric analysis curve that showed that blank microspheres prepared at different emulsification speeds had thermal characteristics comparable to pure PLGA. Both pure PLGA and blank microspheres exhibited the same degradation profile. It's called a single-stage decomposition with a mass loss of 69 until 71%. A stability study was carried out between two formulations. It's F5 with PLGA concentration of 500 mg per milliliter and F6 with 800 mg per milliliter. In vitro stability testing over 28 days showed that the erosion or degradation profile of microspheres at 800 mg per milliliter PLGA was closer to that of the reference product. Therefore, this concentration, 800 mg per milliliter PLGA, was selected for Leuprolide acetate encapsulation. The FTIR characterization showed successful encapsulation of Leuprolide acetate in PLGA microspheres, as the characteristic peaks of Leuprolide acetate were no longer visible in the microsphere spectrum. The infrared spectra of the vinyl microspheres showed high similarity to pure PLGA. It's around 98.44%, and the actual drug loading was 10.6% with encapsulation efficiency of 71.6%. The DSC thermograms of Leuprolide loaded microsphere before lyophilization, as you can see in the figure A, showed a successful encapsulation indicated by the disappearance of broad endothermic peaks between 26 until 55 degrees Celsius and the sharp peak at 166 degrees Celsius. After lyophilization, as you can see in the figure B, with mannitol as a bulking agent, the glass transition of the microspheres remain unchanged and comparable to the reference product, with a mannitol melting peak observed at 162 until 164 degrees Celsius. These are the TGA thermograms. The figure A shows that encapsulation of raw materials, gelatin and leuprolide acetate, in PLGA resulted in thermal changes with thermal decomposition of microspheres occurring at lower temperatures compared to the pure PLGA. While figure B shows that lyophilization with mannitol as a bulking agent led to thermal decomposition of the final product at lower temperature compared to pre-lyophilized microspheres due to the effect of mannitol. And the figure C shows that the final microspheres had thermal characteristics close to the reference product, with decomposition occurring at 303 degrees Celsius, about 10 degrees Celsius lower than the reference. Accelerated in vitro testing was carried out under two different conditions: varying agitation speed, medium volume, and apparatus. The effect of medium volume was observed at the 2-hour point. In 10 milliliters medium with lower agitation speed, initial drug release was higher compared to 1 milliliter medium. The effect of agitation speed was observed after 72 hours when more than 50% of leuprolide acetate had been released and microspheres had undergone deformation and hydrolysis. Higher agitation increased microsphere interactions, resulting in faster release under the second condition. Both in vitro methods produced similar release profiles between the vinyl product and the reference product, but the second method achieved 100% release about 24 hours earlier. Particle size distribution of leuprolide-loaded microspheres was larger than that of blank microspheres due to increased viscosity of the emulsion caused by leuprolide acetate addition. Particle size distribution of drug-loaded microspheres was larger than the reference product but still showed good similarity. Some images during in vitro testing also showed erosion and degradation profiles similar to the reference product. The DSC thermograms of the vinyl microspheres and the reference product showed comparable profiles during dissolution testing, with decreasing glass transition temperature as leuprolide acetate release increased. The reference product showed a glass transition temperature decrease of 7.13 degrees Celsius, while the vinyl microspheres showed a decrease of 7.49 degrees Celsius. And last is TGA thermograms of the final microspheres and the reference product showed comparable profiles with weight loss rate increasing alongside leuprolide acetate release. As the final conclusions, the main conclusion is leuprolide acetate microspheres produced under controlled process conditions exhibited a drug content of 10.6% with an encapsulation efficiency of 71.6%. The glass transition temperature was 49.91 degrees Celsius, comparable to the reference product, and the microsphere showed a similar single-phase thermal decomposition profile. The particle size distribution is also closely matched to the reference product. FTIR analysis confirmed successful encapsulation with a spectral similarity of 98.44%. Accelerated in vitro testing at 50 degrees Celsius demonstrated a sustained drug release profile up to 264 hours, with some observations revealing a comparable erosion or degradation pattern and thermogravimetric characteristics consistent with the reference product. Our findings highlight the promise of PLGA microspheres for long-acting leuprolide delivery, paving the way for future in vivo and clinical investigations. And thank you for your attention.